Hello students, in today's session we will discuss about the pollination and its type, which is being in your syllabus, same 4, paper 1 unit 4. So let us talk about pollination and its types. Now what is pollination? The transfer of pollen grains from anther to the stigma of a flower is known as pollination. Now as we know, the pollen grains are immobile, they cannot reach the stigma by themselves. An external agent is therefore required for this. Now the external agents may be wind, water, animal, gravity or the growth contacts. So after knowing pollination, let us know the type of pollinations. Pollination, it is of two types, self-pollination and cross-pollination. Now as the name suggests, self-pollination, it is also called as autogamy, and cross-pollination or xenogamy. Now self-pollination, the pollination either it may be in the same flower, that is autogamy, or it may be of the different flower belonging to same plant. Now that is known as getinogamy. So autogamy is the transfer of pollens from anther to the stigma of the same flower, and getinogamy is the transfer of pollen from the anther to the stigma of different flower belonging to same plant, that is genetically similar. Cross-pollination or xenogamy, it is exactly the opposite. Now the transfer of pollen grains from one flower to the another flower. It requires abiotic agencies as well as the biotic agencies. These two agents are required for the transfer of pollen grains. Abiotic agencies includes anemophily, that is wind, with the help of wind, and hydrophily with the help of water. When the transfer takes place with the help of water, it is called as hydrophily. Whereas the abiotic agencies includes entomophily, that is transfer through via insects, or charyotophily, transfer through bats, melexophily, that is transfer through snails, and ornithophily, transfer through birds. So let us discuss now what is self-pollination. Now self-pollination, it is the transfer of pollen grains from anther of a flower to the stigma of either the same or genetically similar flower. Now according to self-pollination, it is of the transfer takes place by the means of two ways. First is called as autogamy and the second is getinogamy. What is autogamy? It is a transfer of pollen grains in which an intersexual or perfect flower is pollinated by its own pollens. Now it occurs by two methods, either homogamy or clusterogamy. Anther and the stigma of chasmogamous or open flowers are brought together by growth, bending or folding. That means when the transfer of anthers takes place from anther, when the transfer of pollens takes place from anther to the stigma of same flower, that is homogamy. That means the flower is bisexual and it is open. Whereas the second condition is clusterogamy. In here the flowers are closed. That means no chances. That means every chances, 100% chances of self-pollination, because the anthers and the stigma, they are present inside as the flower is closed. The pollination and seed sets are assured here. Pollinators are not required. That means no external agents are required. See here, the self-pollination, that means the flower having both the sex organs, that is anthers as well as the stigma, that is androecium and the gynoecium, and here the pollination is taking place within this flower only. This second one, it is a closed flower inside which the anthers and the stigmas are there. It is an open flower and it is a closed flower. Then what is getinogamy? It is a type of pollination in which pollen grains of one flower are transferred to the stigma of another flower belonging to either the same plant or genetically similar plant. In getinogamy, the flowers often shows modifications which are similar to one which are found in xenogamy, that is the cross pollination. Now let us see now you might have got the idea what is self-pollination. Now let us see the advantages and disadvantages. Now advantages, advantages includes, as it is a self-pollination, that means the crossing of both the gametes of the same flower, we can get purity is maintained and purity of races indefinitely. That is, it maintains the parental characters or purity of the race indefinitely. Then, self-pollination is used to maintain pure lines for hybridization experiments. The plants does not need to produce large number of pollen grains, and flowers do not develop devices for attracting the insect pollinator. These are the advantages. Now let us see some disadvantages are there, as new useful characters are seldom introduced. That means rare chances are there for new varieties. Vigor and vitality of the race decreases with prolonged self-pollination. As we are taking as there is going self-pollination, it is chances that the progenies after F3, F4, F5, they become weaker and weaker. Immunity to disease decreases, as well as the adaptability to change environment is also reduced. So these were the advantages as well as disadvantages of self-pollination. Now let us see what is cross-pollination. Cross-pollination is also called as xenogamy or allelogamy. Allogamy. Cross-pollination is the transfer of pollen grains from anther of one flower to the stigma of genetically different flowers. Cross-pollination is performed with the help of certain external agencies. Now, various agencies, as you can see here, they are described here. That is either wind or water. Now, wind and water, they are abiotic agencies, whereas biotic agencies includes insects, birds, bats, snails, ants, and snakes. Then what is anemophily? It is a mode of cross-pollination or transfer of pollen grains through the agency wind. That is, examples in the plants, the anemophily pollination is observed in coconut palm, date palm, maize, many grasses, and cannabis. Now, what are the characteristic features? That the flowers are colorless, they are odorless, and they are nectarless. Pollen grains are very light, small, and winged or dusty, dry, smooth, non-sticky, and unbeatable. Stigma is hairy, feathery, or branched to catch the wind-borne pollen grains, and the pollen grains are produced in a very large number. See this, you can observe this in the picture. These are the grasses having this coconut palm, and they are having feathery stigma to catch the wind-pollinated anthers. Then hydrophily. Now it is a mode of pollination or the transfer of pollen grains through the agency, that is water. Example is Zoosteria and Vallisneria. Let us see the characters. Flowers here produced are small and inconspicuous. Nectar and odor are absent. Pollen grains are light and unbeatable due to the presence of mucilage cover, and the stigma is long, sticky, but unbeatable. See here, these are the two types, epihydrophily and hypohydrophily. Epi, you can see the plants, they are on the surface of the water, and here the plants, they are slightly submerged. Then entomophily is another form in which the pollen grains are transferred to a mature through the agency of insects like moths, butterflies, wasps, bees, beetleless, etc. Here the flowers, as you are observing here, the flowers, they should be brightly colored. They are brightly colored. Mostly insect pollinated flowers have a landing platform. Then the pollen grains, they are spiny, heavy, and surrounded by a yellow, oily, sticky substances called as pollen kit. Stigmas are often inserted and sticky. Some flowers provide safe place to insects for laying eggs. Example, yucca. See here, the insects, they are visiting here in the pollen grains. They are having a landing surface, and the pollen grains get stick on either on their body or the wing parts, and they are being transferred from this flower to this. Then comes the next, ornithophily. Here it is a mode of allogamy performed by birds. That means pollination by the help of agents, birds. Only a few type of birds are specialized for this. They usually have small size and long beaks. Let us see the characters. Ornithophilus flowers are usually brightly colored, red, orange, yellow or blue. The floral parts are commonly leathery. The ornithophilus flowers secrete abundant watery nectar or have edible parts. Scent is often absent. Now we are proceeding toward the last part, that is the methods of cross-pollination. Now methods to ensure cross-pollination. There are different types. Let us see dichogamy. As the name suggests, dichogamy. Different types. Anther and stigma matures at different times in a bisexual flower so as to prevent the self-pollination. Then protandry. Here the dichogamy, it is having two conditions, protandry and protogamy. The name suggests itself. Proto means first, andry means the androecium matures first. Anthers mature earlier than the stigma of the same flower. Their pollen grain becomes available to the stigmas of the older flower. Examples are sunflower or the salvia. Then protogamy, it is just the opposite. Stigmas, they mature earlier so that they get pollinated before the anthers of the same flower develops pollen grains. Examples mirabilis, jalapa, that is 4 o'clock plant, gloriosa and plantago. Then the next condition is self-incompatibility. It includes hercogamy and self-sterility. Here, hercogamy means in some bisexual flowers, the structure of male and female sex organs itself proves a barrier to self-pollination. That means either the male flowers are inside and the female organs are on the upper part of the flower, or just the reverse, either the length of that, the difference lies between their length, so that they can be a barrier for self-pollination. Then the next one is self-sterility. The pollen grains of flowers, some of the flowers, they are having pollen grains which do not germinate on the stigma of the same flower. It is because of the presence of some sterile genes, that is S1, S3 in pistil and S1 or S3 in the pollen grains. Example, tobacco, potato and crucifers. And the last one is heterostyly. These are of two or three types. So there are two or three types of flowers with different heights of style and stamens. Diheterostyly, that is dimorphic heterostyly. There are two types of flowers. Pin-eyed, that is having long style and short stamens. Thumb-eyed, that is short style and long stamens. Example, primula. And the third one is triheterostyly, that is trimorphic heterostyle. That is, there are three types of flowers with different heights of styles. Example, Lythrium. Pollination occurs between anthers and stigmas of the same height present in different flowers. So here you can see these are the three types. First one is having, see, the height of the stigma is more than that of the anthers. Here the height is less than that of the anthers, and here see how the pollens and the anthers, they are transferred from here to here. That means the height of the same stigmas, the pollen grains are being transferred. Artificial pollination. Now this technique is used for the purpose of hybridization. It involves pollination of desired female parents with pollens from the desired male parents, taking all precautions to prevent contamination of stigma with undesired pollens. In unisexual plants, the procedure is simple, but it's complicated in bisexual ones. The two processes that takes place in bisexual flowers are emasculation and packing. Here the steps are being reviewed here. That is the stigma, and these are the anther, that is the male reproductive organ. And see here how that, from here how the transfer is being made. Then what is the importance of cross pollination? You see, cross pollination, as we know, it is a transfer transfer of pollen grains from anther to the stigma of another flower. So it introduces genetic recombination and hence variations in the progeny also. Cross-pollination increases the adaptability of the offspring towards the changes in the environment. Then it makes the organism better fitted in the struggle for existence, and the plant produces through cross-pollination are more resistant to diseases. Some of the disadvantages are a factor of chance is always involved in cross-pollination. It is less economical. Then some undesirable characters may creep in the race, and the very good characters of the race are likely to be spoiled. Now, these are some of the disadvantages. Now let us see co-evolution of flower and its pollination species. Co-evolution is the evolution in two species that interacts extensively with one another so that each acts as a major force of natural selection on the other. When one involves a new features or modifies itself, the other involves new adaptations in response of it. This constant mutual feedback modification between the two species is known as co-evolution. The co-evolution of the flower and its pollinator species are tightly linked with one another. These are some examples. Deza Navia is a rare orchid found only in a few places in South Africa, and until Johnson came to study it, no one knew how it was pollinated. After a lot of patient orchid watching, he and his colleagues discovered that it is visited exclusively by the fly shown in the figure. This one. Its proboscis is well matched to the length of the orchid, and the orchid grow pollen in just the right place so that they get stuck to the fly. You can see them in this picture, the two dangling yellow pockets on the fly's snout. This fly is named as orchid fly. Now these are the dangling pockets in which the pollens are being attached when it visits the flower. So, and lastly, thank you. Hope you have already